One, two, three, four. Can you hear me via Zoom now? Okay, cool.
If you are okay, I can start to share your session. So welcome everyone from the second part of our meeting, and now we will have a presentation from Martin Steinhaus. He is working in Leibniz Institute for Food System Biology at the Technical University of Munich, and his research interests are mainly on identification of K odorants, odor-active organosulfur compounds in foods and their analysis, molecular causes of flavors in foods, and molecular causes of odors in non-food materials. And today, Martin will present us about the K odorants in foods, identification and use for targeted product improvements. Martin, the floor is yours.
Thank you very much. I'm very happy to be here in Porto and show you a little bit about our research on key odorants in foods. And what we do has actually a lot to do with, no, come on. I think it doesn't work with the, I have to go back. Yeah, now it works. What we are doing has a lot to do with consumer expectations, and you probably identify this scene. It's a scene from the Bible when Jesus fed the 5,000, and it's said to happen with just 5 loaves of bread and 2 fish. And after that, everybody was satisfied. That were consumers 2,000 years ago. If you have consumers from nowadays, probably this would happen. And this illustrates that consumers have a lot of different expectations nowadays. And of course, this was a target of more detailed research. And this is illustrated here on this slide. That was a survey that has been done 10 years ago in Germany. And you see people have a lot of expectations, a lot of demands towards food. But the important thing for us is the top answer is that food shall be tasty. So if food is not tasty, it has no chance on the market. And this is very stable even over the years. This is another survey that is from the United States, and it's repeated every year. And you see, taste is always the top answer, the top driver of consumers' food selection. But honestly, taste is here used in a sense where it stands for the overall sensory perception, not only targeting at the sense of taste. It includes all our senses that are involved when we consume food, even audition, if you think about crunchy potato crisp, for example. Vision is important before we take the food into the mouth. And when we have the food in the mouth, it's basically the skin senses, mechanoreception, thermoception, nociception, but also proprioception, and then, of course, the chemical senses, gustation and olfaction. And as a matter of fact, among all of these, olfaction is the most important. And I think that all of you already made this experience when you have a cold and your sense of smell is temporarily out of order. Everything tastes very, very boring. And the reason why olfaction is so important, we can nowadays easily explain when we look into the physiology of the sense of smell. Basically, there are two ways that odoractive compounds are perceived during food consumption. This is before the food is taken into the mouth with the inhaled air. This is the blue arrows here. Yeah, the food stands in front of you and you smell the freshly cooked meat, for example. And then it's when you have the food in the mouth and it's chewed. Actually, at that moment, you usually don't have any perception, but when you swallow, after you swallowed, there is exhalation. This is a reflex. And with this exhaled air, the major part of the volatiles that are responsible for perception of this so-called retronasal odor perception are transferred into the nose. And here at the upper nasal cavity, we have actually the location of the sense of smell, and it works basically the same way whether the odorants are transported orthonasally with the inhaled air or retronasally with the exhaled air to this spot. And what happens there is that odorants bind to odorant receptors. These odorant receptors are included in these hairy structures, the so-called cilia of the odorant neurons. And when this interaction takes place, there's an intercellular reaction cascade starting that finally leads to the depolarization of the membrane, and this is then transferred to the brain as an odor information. So when talking about human olfaction, we have to consider three important points. That is that humans have around 400 different of these odorant receptors, that each and every odorant receptor has only one type of these odorant receptors. And third, that all the axons coming from the odorant receptor cell with the same odorant receptor, they are wired together. They meet each other in the olfactory bulb, that is the lower level of the odor information transformation in the brain, in a so-called glomerulus. And so on the level of this olfactory bulb, all the odor information is packed into a kind of pattern that consists of 400 individual information bits. And this is the reason why the sense of odor is so complex and why it's so important for the overall sensory impression of food. And it also shows us, how to analyze these compounds. So what we have in a food basically is this situation. And you probably realize that you saw this similar scheme already this morning. And of course, we have among the food constituents a lot of non-volatile components. They come not into question as being odorants because they cannot make the way into our nose. So odorants need to be volatile to get into our nose with the exhaled or the inhaled air. And so volatility is an important prerequisite for being an odorant, but it's by far not sufficient. So most of the volatiles that appear in a food are not odor active. So to be odor active, they have to have the potential to interact with these odorant receptors, which is illustrated here. And even then, it's also a matter of the concentration. So this was already discussed this morning. Each volatile has odor threshold concentration. And if you look into these odor threshold concentrations of different food odorants, we see the basic problem, or one of the basic problems in odorant analysis, and this is the fact that these odor threshold concentrations are extremely diverse. So the red names here are names of odorants, and you see that their threshold concentrations indicated here by the y-axis, they are extremely different. They cover a range of more than 10 orders in magnitude. If you look at the blue compounds, these are taste-active compounds, so the range is much smaller. And the greens, these are chemostatic compounds. So this is something that is particularly a point when it comes to odorants, that their odor threshold concentrations are extremely different. So that particularly means that some of them are extreme trace compounds. Okay, so this is the differentiation of the volatiles and the odor active compounds. And then we have another point that is particularly a typical case when it comes to foods. In foods, we usually do not have a single odor active compound, but we have a lot of odor active compounds at the same time. So we have mixtures of odor active compounds. And it has been shown in sensory experiments that if you have mixtures of odor active compounds, not all of them contribute to the mixture, to the odor of the mixture, even though they might be present in a concentration above their individual odor threshold. If it comes to a mixture, some of them are suppressed by more potent ones, and so they would not contribute to the overall mixture. And those who do contribute, we call the key food odorants. And they are not very big in numbers. Typically, you can reconstitute food aroma with a number of 10 to 20 compounds. Sometimes even you need just a handful of compounds, and everybody recognizes the aroma as the aroma of a certain food. And yeah, these key food odorants, these are our target compounds. And as I already mentioned, this scheme also gives us the idea how to analyze, how to identify these key food odorants. And the first step would be the differentiation of the the non-volatile food constituents and the volatiles. And this is typically done in a preparative way. So we simply separate the food constituents from the volatiles. And we already heard today that there are modern methods that do not use a lot of organic solvents, which is a big advantage. But when we are targeting at a screening of odor-active molecules in food, we still use organic solvents. And the reason is we have to try to avoid artifact formation. Artifact formation is a major problem if you're interested in food odorants. And the reason is that food typically does not only contain odor active compounds, but also their precursors, particularly precursors that are converted by thermal reactions into odorants. So what we have to avoid is any kind of heat. And the general approach that we use is we cryomill the food, typically done at the temperature of liquid nitrogen, and then we extract the food with an organic solvent, with a low boiling organic solvent, such as diethyl ether, for example, or dichloromethane. This is done at room temperature or a little bit below room temperature, particularly when it's aqueous food. We add typically also some anhydrous salts, and we have to make sure that the solution heat is not generating higher temperatures. And then we have the most important step, namely the separation of the non-volatiles from the extract. And this we are doing with a very specific method, a method that has been developed in our institute. The first publication was 20 years ago. In the meantime, it was further developed, and the last basic improvement was that the manual instrument was converted into an automated instrument with a pneumatic valve, and now fully functions automatically. What is the idea? The idea is that small portions of the extract are allowed to enter high vacuum, where there is a very quick volatilization of the volatiles. The non-volatiles stick to the glassware. The volatiles are going through the apparatus, and any kind of non-volatile material that is in there as small droplets is deposited at the walls. This apparatus is under high vacuum to make sure that the volatiles have a high vapor pressure, and finally the volatiles are recovered in a cold trap cooled with liquid nitrogen. Normally we do this with an extract, with a solvent extract, like shown here on this slide. If we have aqueous food materials, beverages particularly, we can also directly apply it, and this is normally not done because water is not as volatile as a low boiling organic solvent, but it can be done. And this is good to show why this approach is so successful, why it or that it doesn't generate artifacts. And I apologize to all people connected via Zoom, because they cannot have this feeling now by their own. But I have brought here a beer distillate, and if you like, you can distribute it to the audience. And this is the most important thing during our research. At all points, we have to make sure that we get representative isolates. And the best way to do this is simply to use our nose and make sure that our distillates, that our extracts, that our concentrates actually represent the aroma of the starting material. Okay, this was the first step. The next step is now differentiating the volatiles from the odor active compounds. And this is done by GZO olfactometry. That means we use a gas chromatograph with a column, and at the end of the column, the effluent is divided. One part goes to an MS or an FID. This is the simplest version, and another part goes to a heated exit, the so-called sniffing port, which allows us to localize the odor active compounds in the chromatogram and characterize them by their odor description and by their specific spot in the chromatogram, by their retention index. Again, it's important that we minimize the heat impact during GZO, and particularly I want to stress the importance of the injector in that field. So we do only cold on column injection to avoid heat impact during injection, and we were able to show recently that hot injection can lead to artifact formation. Of course, compounds would not be degraded to 100%, but even if they are degraded by 5 or 10%, if the products are highly odoractive, you can end up with a completely different picture of a GCO result, and this needs to be avoided. So cold on column injection is a major point if you want to do a representative screening of odorants. So what we typically do when we do GCO is to dilute the sample and prepare a dilution series. And we repeat the GCO analysis on this dilution series. And then you can imagine the higher the dilution, the lower is the number of compounds you would detect by GCO. We typically go on until there's no odorant detected anymore in the whole run. And we define the dilution factor of that dilution, where this compound, the highest dilution, the dilution factor of the highest dilution in which this compound was detected, then as the flavor dilution factor of this compound. And this has two major advantages. The first one is we can easily compare two samples or even more samples on this early level of the investigation by comparing their FD factors. And one of the examples shown here on this slide, this is the analysis of a cocoa sample with an off flavor, with a smoky off flavor. And as a reference, there's a cocoa with a flawless aroma. And this enables us, before we even know what compounds are behind these odor impressions, to find out which are or where are in the chromatogram are the compounds that could contribute to this odor difference by looking at this, at the number of the flavor dilution factor. Yeah, you see here we have these smoky odor notes on the one side, and we clearly see that in the smoky cocoa, the FD factor is higher, or even some of them, actually most of them, are even not detected in the reference cocoa. But even for that one, we see there is a difference in the FD factor, and this is higher in the smoky, in the off-flavor cocoa than in the reference cocoa. So it's always good for comparison. Yeah, it's not only if you have an off flavor, but also, for example, it could be a gold standard of a fresh fruit in comparison to a processed fruit product or something like that. The other important information that is generated from FD factors is we can already talk about the relative importance of the individual compounds. Yeah, the higher the FD factor, the more or the higher is the probability that it is actually a key compound. That means we can, particularly with the time-consuming step of structure identification, concentrate on the compounds with high FD factors. At least we can first concentrate on them and see if we succeed with them. And in a later step, if this is not confirmed, we go down a little bit. And so we can step by step include more compounds until we finally succeed with the identification of the key compound. So I already mentioned the structure assignment is often very time consuming. And we heard in a talk this morning about GCO-MS. So you can, of course, do GCO in combination with MS, but this doesn't guarantee that you can directly identify the compounds that are responsible for the odors you perceive during GCO. Honestly, in most cases, it won't work. And the reason is, of course, the diversity of odor thresholds and the compounds, so in the odorants. So we often have trace compounds that are responsible for the odor. Trace compounds, you have no chance to get a mass spectrum, either because the sensitivity of your instrument is not sufficient, or more important, you have co-eluting compounds that are present in higher concentrations, so you would never get the mass spec of your odorant, but you get the mass spec of a kind of matrix compound. And this is the reason why, when we are interested in the odor active compounds, in the identification, we typically go first for the retention index and the odor characteristic, and go into databases and see if we can do a preliminary structure assignment with these two parameters. And of course, you need reliable data on that. And yeah, we have addressed that also in the talk before the lunch break. That it's important to have these databases, and we have set up a database since 20 years, and since more or less 1 year now, this database is available free of charge on the internet. So it's called the Leibniz LSB Atom Odorant Database. So you can, for example, type in the odor quality and an approximate retention index, and then you will get back the compounds that have this word in the odor description and a retention index in a certain range around this retention index you typed in. Nevertheless, this is always helpful to get an idea. If you have a compound found that is probably fitting, then of course you would have to get the reference compound, analyze it by GCO, and the final structure assignment, of course, needs GCMS. Of course, if you come up with a new compound, you also need to go to GCMS and try to get an idea on the identity of the compound first by GCMS. And then you have the problem of avoiding matrix components. And the only way you can do that is by fractionation, activity-guided fractionation. That means you first fractionate your extract, for example, by acid-base extraction, by column chromatography, and this is the most sophisticated way, and this is shown here on that slide, by two-dimensional gas chromatography. Of course, if you do fractionation, you always use GZO. This is the activity-guided thing in the whole approach to localize the target compounds in the different fractions. And in the case of two-dimensional gas chromatography, that means you have a sniffing port on both dimensions. So what you would do is you would first inject your extract, you distillate, you concentrate on the first GC, would separate it on a column, let's say a polar column, and then you determine the retention time of your target compound. This is the first run. The second run, then you would select the window in which the compound eluted in the first run, and have the eluate transferred to the second oven. The second oven, you do a refocusing, typically with liquid nitrogen, and then you send the compound for further analysis to a second column, of course, a column of a different polarity. So if you start with a polar column, you'll probably have here a column, the unpolar column or a middle polar column. And then you can do sniffing again in the second dimension, so you can directly see at which point your compound elutes, and then you combine that with a mass spec, and then you can actually get the mass spectrum of the compound. So we do that since a couple of years now with a high resolution mass spec, which gives us even more information. And this is what we did, for example, for the identification of the compounds responsible for the smoky off flavor, and we ended up with 7 phenols that were present with higher flavor dilution factors in the off flavor sample than in the reference sample. Okay, that was the differentiation of the volatiles from the odor-active compounds, but we are still talking about odor-active compounds in our extract. We still do not know if these compounds are odor-active in the starting material, in the food sample, and whether they are actually contributing to the overall aroma of this food. And to find out this, we have to do sensory experiments. There's absolutely no way to predict which of the compounds in the end would contribute to the overall odor. So the only way to find out is to do sensory experiments, and the basis for these sensory experiments is the exact quantity, the exact concentrations of the compounds. So the next step is the quantitation, and we typically go for the gold standard in analytics, stable isotope dilution analysis. So we use stable isotopically substituted analogues of our target compounds as internal standards. So these would be added at the very beginning of the process. We homogenize the added standard with the material to achieve equilibrium between our analyte molecule and the added standard molecule. And once this equilibrium has been achieved, any losses that we face, even if we have sensitive, if we have reactive compounds, we can even lose 95%. It won't change the ratio of the analyte and the standard. And this is what we finally determine by GC-MS analysis, typically in CI mode, chemical ionization mode, to make use of the different molecular mass of the analyte and the standard. Yeah, the big advantage is, as I already mentioned, that any losses are fully compensated, but the price we pay for that is that for each analyte we need an individual standard, and some of them can be easily obtained by simple synthetic approaches like this ethyl butanoate. This can be obtained by simple esterification. But a compound that you are all very familiar with, jasmine, is one of the examples that require quite complicated synthesis, and this is one that we published the last year, where we incorporated 3 deuterium atoms in the jasmine molecule within 8 steps and 16% overall yield. I don't go into details, just as an example of a rather complicated synthesis. So once we have the concentration data, the next step is to compare them with the other threshold concentrations. And a very easy way to do that is simply to divide the concentration by the odor threshold and get what is called the odor activity value, which is simply the factor the concentration exceeds the odor threshold. That means odor activity value above 1 means the compound could contribute to the overall odor. Compounds with odor activity values below 1 typically do not contribute to the odor, to the overall odor or aroma of the food. So this is what we would do next. We would exclude them. This already reduces the number of compounds. In this case, this was research on guava fruit, where we now have the problem limited to 13 compounds with odor activity values above 1. And with these compounds, we then go into the proof of success stage, which means we do a reconstitution experiment. We try to reconstitute the aroma. In this case, we use these 13 compounds in the natural concentrations in water and compared the aroma of this reconstitution model with the aroma of the fresh fruits. And as you can see from these spiderweb diagrams, it worked very well. Disadvantage of this guava fruit reconstitute is that it's not really stable. And so again, I apologize for all connected via Zoom. I have a reconstitute with me. This is unpublished results. This just came out recently from our research, and it's a reconstitution model of walnut. Be careful, it's in silicone oil, so it's a bit fatty. It's recognizable.
Yes, very well.
Yeah.
Very well.
Yeah, I was also very satisfied with that one, to be honest. Yeah, so this is the important thing. We need the proof of success, and then we can finally go to omission test. That means we omit individual odorants from this full reconstitution model and see whether a difference is obtained. And if there is a difference in a 3 AFC test, 3 alternative forced choice test, then we can consider this particular compound a key odorant. And as you see here, this even happened not for all of these 13 compounds, just for 8 of them. And these 8 compounds we can finally define as the key odorants. So as I said, in the end, it often turns out to be only a few compounds, 10 to 20. In this case, it was only 8. And these, of course, are then major quality parameters, major substances responsible for the sensory quality of these guava fruit. So altogether, it's a 7-step procedure. It starts with the mild isolation of the volatile, screening by GCO, then structure identification of the most potent odorants, then quantitation, calculation of odor activity values, the proof of success by odor reconstitution, and finally omission tests to find out which are the key odorants. So what are we doing that for? Of course, it's always nice to find new compounds. This is part of our research. It's more like basic research, or you might call it curiosity-driven research. This slide shows a couple of tropical plant food materials. We were able to find new odor-active compounds that have not been appearing in the scientific literature before. But of course, our main target is more in the field of applied research. What we do is we monitor these key odorants along the food value chain, mainly more or less around the food itself. So particularly we study processing and aroma changes during processing on the molecular level, also of storage. But we also, in the recent years, we go more and more upstream into the influence of agricultural practices. And we have currently the plan for collaborating with breeders, so to use targeted breeding for specific plant materials with a good aroma. And we also study to the more downstream part of the whole thing. For example, what happens during a consumption. It's interesting that particularly for dry foods, many odor-active compounds are not in the food itself. They are released not before the food becomes into contact with the saliva. And together with our biologists in our physiological department, we study also the perception of the compounds in the nose. A major task is definitely the influence of processing, and the approach is shown here. So typically we have a raw material. If it comes to fruits, for example, the raw material is often the gold standard, the fresh fruit, and the product has probably problems, different kind of aroma that is not considered the optimum. And so what we do is we identify the key compounds in the raw material and in the product. So this shows us which compounds are responsible for the aroma difference. Then we quantitate these compounds during the individual processing steps, which then shows us which are the crucial steps during the processing. And finally, we can concentrate on these steps, try to optimize them with the aim of a product with a better aroma in the end. So 2 examples on that. This is actually a project that I did in my postdoc was about pepper, and particularly white pepper shows sometimes a kind of fecal off flavor, and it was hypothesized that this is not present in black pepper and green pepper, and so the question was, has it to do with ripening, because white pepper is produced from ripe pepper, or has it to do with the processing, particularly the fermentation? And we applied our approach. We identified 3-methyl-indole and 4-methylphenol as crucial compounds responsible for this off flavor. And then we quantitated them, and we showed that fermentation is actually the crucial process. This was the fermentation process we found on site. So you see relatively high concentrations, relatively quick starting of the formation of these compounds. And we tried to find better solutions, adjust the process, and in the end, it turned out that the water throughput during the fermentation process is the crucial point. If you have higher water throughput, we end up with a lower formation of these off-flavor compounds, and also we can even prevent the formation of these compounds for a longer period of time, which allows for different techniques to avoid the compounds being offensive in the final product. It's not all about off flavor. This is shown here. This is data from another project about apple juice. Again, we identified first the crucial compounds, and one of the most important compounds in apple juice is the E2-hexanol, which has a very nice green apple-like flavor. And what we showed is that during apple processing, it's basically 2 steps that are crucial. That's the crushing, and that is the final heating step during pasteurization. These 2 steps make basically the aroma changes, and the E2-hexanol comes from the crushing. It's a lip oxygenase reaction that happens during the crushing that leads to this product. It's an oxidative reaction. And the problem the juice industry faced for a long time is that they tried to avoid enzymatic browning. So what they did is they kept the time between crushing and pressing as short as possible. And also what they did is they added ascorbic acid. So this led to a juice with a nice color, a nice bright color, but at the same time, it also prevented the oxidative formation of this important flavor compound. So in the end, we had to tell the juice industry, you have to decide whether you want a nice flavor or a nice color. This slide just shows that it's not all about food. You can apply the approach to any kind of material, and particularly we are at the moment doing a lot in the field of car components, polymer materials, and this is one example, one of the older projects on car components, where you see a couple of amines used as catalysts were identified as crucial compounds. And with this knowledge, it was possible to improve the initial product, where you can see here this big peak was the crucial compound. And after our research, the optimized product, this compound was not present anymore. And this is also somehow related to your topics, to water, because what is basically the difference between food and water is in food. The gold standard is not the odorless food. It's typically the fresh fruit or the traditional food, or the handmade food or whatever, but it has odor-active compounds. They are wanted, they are quality parameters. Here in this kind of research, like in water, it's typically the odorless material that is wanted. Okay, if you're interested in further reading, There is a nice book on gas chromatography that came out last year, and this holds a chapter written by me on gas chromatography olfactometry, and there you can find more detailed information. And yeah, now I'm open for discussions. I'm happy to take questions, and I'm curious how we could contribute to the WaterTOP problems. I mean, I hope I could make clear what our basic expertise is. It's the identification of new odor active compounds, particularly the generation of representative extracts, also for the screening purposes. And yeah, the other expertise we have is the synthesis of isotopically substituted compounds that might be also helpful for water analysis in combination with GCMS. Thank you for your kind attention.
Thank you, Martin, for your very nice and informative presentation together with the odors of walnut and beer. We have a little bit limited with time, and so I can have 2 questions, very quick ones, if there is, from the audience. I think there is no. Yeah. So if there is no questions, I think we were really very watch your presentation very interestingly and couldn't think about the questions more.
Yeah, if there are questions up, please feel free to contact me by email. I'm very happy to answer them by email as well.
Yeah, thank you very much, really. So